Hello, I'm Dr. Ahmed Abdelal, an associate professor of communication sciences and disorders at Bridgewater State University. And this is an introduction on the cellular structure and functions of the nervous system, with a special focus on the brain and the neurotransmission. Okay, so the human nervous system is made of two main divisions. We have the central nervous system, that is the brain and the spinal cord, and then we have the peripheral nervous system, which is all the nerves that come out of the brainstem and out of the spinal cord. So we will speak about this later, but now we are going to speak about the cellular composition, the cellular structure of the brain and of the nervous system generally. So the brain is about, for the adult, is about 3 pounds, and in the newborn it's less than 1 pound. Generally, on average, it is 0.8 to 0.9 of a pound in the newborn. And because human beings are born with generally the brain is small, and about 1/3 of the size of the adult brain, that gives the environment the opportunity to sculpt the brain and to connect it and to develop it in a way that will make the human being a best fit for the environment that he or she lives in. So even though it makes, about 2% of the body on average, the brain consumes a lot of energy, and it doesn't store energy. The energy is basically taken out of glucose, oxygen, and some other chemicals that the brain takes from the bloodstream. However, it doesn't store food. It doesn't store nutrition. So in the adult human being, the brain consumes about 15 to 20% of the nutrients. Everything that you consume, 15 to 20% goes to the brain. And in children, in newborns, up to 50% of what they consume is dedicated to the brain. So it uses electric currents, and you could say that in the entire nervous system, your brain, spinal cord, and the nerves that come out of them, all of the nervous system, gives you about 40 watt electricity, which is the electric energy inside of your nervous system is equal to the light bulb that you have in your fridge. So when we look at the cells inside of the nervous system, there are two kinds. One kind is the neuron itself, and that is bigger, and it transmits signals, across the body. So for example, one neuron can transmit signal to another neuron. One neuron transmits a signal, say, from your forehead to your toe, for example. So it sends signals that go from one part of the body to another. But the glial cells is another category, another kind of cells, and these cells, they kind of support the neuron in different ways, and without them the neuron cannot function at all. You can think of the relationship between neurons and glial cells as, say, for example, when you order something from Amazon or somewhere, it comes in a box. Let's say order a textbook, and you have the thing that you order, and around it there are a lot of peanut foam kind of things that are cushioning that thing that you are sending, so that to protect it and prevent it from being damaged as it is traveling. So the glial cells, the name glia means glue from Latin, and there are more than there are so many kinds of glial cells, and they outnumber neurons by 50 up to 50 times. Like for every neuron, you have almost 50 glial cells. So they cushion the neurons. They do a lot of functions to enable the neuron to perform the neurotransmission part, and in the best way. So collectively, these glial cells, they provide insulation for the neuron. When you insulate, you increase the speed of transmission of signals. And then also they provide support in different ways, like giving nutrition. They regulate neurotransmitter activity in the brain and the nervous system. They contribute to the development of the brain as babies are developing their brains. And they also enable us to form the blood-brain barrier. So we'll explain this, but these are the general functions, generally speaking, about all glial cells. And some of these glial cells, some only, not much, just a very, very small fraction, are able to transmit signals. So as we continue, you need to take good notes, because at the end of this lecture, you are going to have some questions that will, kind of like a quiz on the information that I'm discussing here. And if you cannot answer these questions at the end, then you need to go back and to listen to the lecture again, or to that part of the lecture again where you can get the answer. I also would recommend that you pause the lecture every maybe half hour or so, check your information, look at your notes, take a break, and then come back later so that you can maximize what you will get out of it. So now we are going to speak about 5 different kinds of glial cells. And after that, we will be talking about neurons and how neurons transmit signals. So the first kind of glial cells are astrocytes. The word astro in Latin means star. Cyte means cell. So astrocyte means the cell that looks like a star. And you can see it in green here. This kind of cell provides or plays at least 4 or 5 different functions. So first, it has, as you could see, arms, these extensions here, and it has actually feet called pods. So with these arms, what it does is it makes connections between a blood vessel on one end, and it makes connection, on the other side with neurons. So it has little like structures called pods or feet that stick and kind of connect with neuron walls of neurons. And by doing so, it provides support. It provides structural support. It connects two things and kind of minimizes their movement away from each other. So, in addition to that, as it connects to blood capillaries, blood vessels, it establishes through these little feet or pods, and between them and between the blood vessel wall, it establishes a barrier called the blood-brain barrier. So as it is mentioned here, the blood-brain barrier is a highly specialized kind of structure where the these little pods come in contact with the walls of the bloodstream and the blood vessel, and they have little, channels in between. And the astrocytes extract from the bloodstream the nutrition that is needed for the neuron. So what it does, it takes the substances exactly as they are needed and in the amounts that are needed by the brain, and it passes them on to the neurons. So here you have a blood vessel on one end, and then you have a neuron, say axon on another end. So, and by doing so, it enables the brain to be protected from the dangerous substances that could, that are in the bloodstream that could damage and kill neurons. So that is called the blood-brain barrier. And you would notice that in some medications, and frequently, they would mention, for example, medications for ADHD or depression, or they will tell you if they penetrate the blood-brain barrier. So one substance that can be kind of force itself through the blood-brain barrier is alcohol. It just seeps through, and this is how it makes a person lose control over their mind. So that is another function, and you can imagine if there is deficiency in nutrition, then the neurons cannot fire, cannot do their job optimally. In addition, the astrocyte cells also regulate neurotransmission amounts. Cells, when they interact with each other, they use neurotransmitter molecules that go from one neuron to the other, and in doing so, they channel the information from one neuron to the other. So we use potassium, calcium, sodium, and different chemicals, and particularly potassium. If there are more potassium molecules out there than needed, the astrocyte could absorb the excess and either send it to another neuron that needs it, or it can hold onto it until it is needed again. So it regulates synaptic activity by kind of controlling the amount of neurotransmitters that are available out there. And it also recycles some neurotransmitters, because not all neurotransmitters are broken down. Some of them are broken down when they are used, and then they are recycled. So that cell plays a tremendous role in the maintenance and nutrition of neurons, and also in their ability to transmit signals through control of neurotransmitter substances, especially potassium. Another kind of glial cell that is number 2, that is the Schwann cells. Schwann was someone's name, a scientist, and these are named after the person, Schwann cells. So a Schwann cell, you can think about it as a segment of marshmallow, but it is very, very tiny. Now, it's very important that you know exactly where the Schwann cell resides, what part of the nervous system it works in, and also how it works, how it is made. Because you have another cell that is similar to Schwann cell, but it is, in function, but the structure and the location are different. So Schwann cells make myelin, make the insulation material for the axons of neurons. However, Schwann cells make myelin in the peripheral nervous system, in nerves that come out of the brain stem and out of the spinal cord. So, for example, this is a motor nerve. And as you could see, it has an axon that has little insulation segments that are like pieces of marshmallow. These are actually long sheets that are wrapped up to 16 times wrapped around the axon. So like like that, one segment of the axon. So one little segment, as you could see it here, is a single Schwann cell. And you need many Schwann cells to insulate the axon of a single neuron. That's a very important difference. So a Schwann cell, again, exists in the peripheral nervous system. It makes the myelin of the peripheral nervous system, like your arms, your trunk, your legs, and you need multiple segments to insulate a single neuron, for example. Any of that insulation substance, the white substance that's insulating the neuron axon, is called myelin. So that is the substance itself. And the segments that make the myelin of the peripheral nervous system neurons, these segments are made of Schwann cells. They are actually Schwann cells. So if you can count here how many cells in this neuron axon, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, for example, here. But this is just a kind of an example. Some of the neurons have axons that need 400 segments, 400 Schwann cells to just insulate a single axon of a single neuron. When we say insulation, when we insulate an electric wire, for example, we basically protect the signal from dissipating, from being lost. And if the insulation is not efficient, then the signal will not travel fast, and it will not travel fully, or will not travel at all. So insulation is critical. And if there's degeneration, for example, in these, or the person doesn't have enough layers of them, or there's degeneration, then the person will have a neurological disease like ALS or Parkinson's disease, and so on. Myelin is also made by another kind of cell that is called oligodendrocyte. The name is long, but if you break it down, it will be quite easy. Oligodendrocyte. So oligo is a kind of sugar molecule. Dendro is a branch, and cyte is a cell. So this is the blue here, made in blue, and that is just a small description of it. It makes myelin segments. However, it makes only the myelin segments in the brain and in the spinal cord. Remember, Schwann cells, they only make the myelin segments in the peripheral nervous system. But these in oligodendrocytes make the myelin segments in the central nervous system, which is the brain and the spinal cord. And in addition, how they do that is totally different. Oligodendrocytes have many branches, and they send these little branches or arms to different axons. Like imagine I'm sitting here and I have, like, say, 100 wires coming down from above, tied and coming down from above. And I have, say, 100 arms. And each, I'm going to take one arm and hold on to one axon, and another arm to another axon. And so on. So I have all these 100 arms and holding on to these axons, these little wires that are coming from above. So by doing so, I actually I anchor these neurons. I support them so that they don't go around. And so I support them, giving structural support. In addition, I actually provide the myelin segments for multiple neurons. So one oligodendrocyte provides myelination or myelin segments to multiple neurons, and also the oligodendrocytes are found in the central nervous system neurons. In the central nervous system, and they serve central nervous system neurons. So they provide support, and they provide myelin or insulation. Now, you should be able to compare point by point, compare between the Schwann cells in structure and in function, and to compare this with the oligodendrocytes in structure and function. If you feel you can't do that, then go back and look at listen again to what I said about the Schwann cell and about the oligodendrocyte. So you compare point by point, where each one exists, how each one is formed, what it looks like, and how exactly it makes a myelin segment. The other kind is called, again, we're still speaking about glial cells. This kind of glial cell is called microglial phagocyte. So phago comes from the verb phagen, means to eat. Cyte means cell. As you could see, this is a cell, for example. When it sees a bacterium or some debris, like a cell is destroyed, it has little debris, little pieces broken down and sitting there and the space between cells. Then the microglial cells, they kind of extend themselves and they distort their shape and they engulf, they eat these, parts, either germs or they eat broken parts of cells. They engulf them. You could see that here in this diagram where the bacterium is here and then the microglial phagocyte eats it. It just goes around it, surrounds it, and then it changes its own structure so it can actually swallow that bacteria and kills it. It recycles the debris all around. It maintains the health of the nervous system. And if there's damage or injury, you have a lot of these microglial phagocytes going around to maintain the site and to clean up and to try to protect the site, from further damage. Another kind is called radial glial cells, and these are glial cells that make like rods. So a rod is like a cylinder. Imagine you have like a little tube. So one single radial cell is like a little tube, a little cylinder that is opened from both ends. And then you have another tube and they fuse together. They make a longer segment. And then you get so many of them together like this to actually they connect to make hoses, like little hoses or pipes that go from the site where the neurons are born to the site where the neurons are going to stay throughout their life. So for example, let's say the neuron is born, say, deep in the brainstem, and it is supposed to travel to the forehead so it can be there to help you with memory and to help you with decision making and so on. How does it go there? How does it know exactly where it is going to connect and be embedded in the pathways there? So radial glial cells make like little threads, little tubes that travel from the site where the neurons are born to the site where the neurons will be embedded and stay there for their lifetime. And as the neuron is born, as soon as it is born, it begins to migrate, and it clings on to the radial glial cell, that structure, and it continues to travel like climbing up a rope. As it continues to travel, it makes, through the connections between its walls and the walls of the glial cell, radial glia, it has little channels where it can absorb nutrients, and it continues to grow as it is traveling. So you could see now it is traveling, becomes bigger. By the time it reaches its destination, where it is going to be embedded then it is a fully grown neuron, and then it knows its location, and at the same time it is fed and nourished throughout the immigration process. So radial glial cells, again, are critical to cell migration. They guide neurons to their destination. In addition, they feed neurons, while newly born neurons, while these neurons are traveling or are migrating. So now, two major functions of radial glial cells that I spoke about. So after this, we spoke about these 5 kinds of glial cells. Now we are going to speak about the neurons, the structure of neurons, and their functions. Let's first speak about the functions, physiology of neurons. So based on physiology, based on their function, what kind of job do they play, we only have 3 kinds of neurons. Again, I'm speaking about function, not anatomy, not anatomic structure. So there's one kind of neuron called the sensory neuron or the primary sensory neuron. Either way, you'll find the 2 names, either sensory neuron or primary sensory neuron. And another kind is the motor neuron or the primary motor neuron. So the primary sensory neuron, its function from the name of it is to bring in signals from different parts of the body outside, like your skin, your blood vessels, or inside, like your blood vessels, your stomach, whatever. So to bring in signals, signals like say pain, temperature, different signals that the person encounters, and they channel these signals quietly. They channel these signals up to the brain so that, or the spinal cord, so that they could be processed. Primary sensory neurons are also called afferent neurons. Afferent, you can think of it, the word affect. Affect means kind of the expressions on a face. So what is your affect? Maybe you are smiley, you are, showing anger. Maybe you are, sad. Or maybe just, we call it a flat affect, like nothing. You don't do much. No one can read your face. You just a plain, like a mask face. So afferent, again, it is a different name for sensory neurons. Afferent neurons are sensory neurons because they bring in information, sensory information, from outside or inside the body to the spinal cord and to the brain. Motor neurons. Motor neurons are the neurons that enable us basically to perform movement, whether involuntary movement or voluntary movement. So, especially voluntary, these are called efferent. Efferent. Remember from the word effect. Effect means the consequences of something. Something happens and it makes an effect. So please make a difference between affect or affect and effect. They are totally different words, but students sometimes confuse one for the other. So efferent has to do with motor, and so for motor, you have the idea to move, and remember, when we speak about motor from now on, we're going to speak about voluntary movement. So you have the idea to move. The idea comes from here, and then finally the signals are processed and then go down to the spinal cord, to the brain stem and the spinal cord, and then you can move hands, you can move lips, you can move any structure that you can think about. So the idea of motor neurons is that they come from higher levels of the brain, because we speak about intentional movement, voluntary movement, and they travel down to lower structures. So they make an effect. We call them efferent neurons. And they innervate muscles, and muscles then are connected to the structures that they actually move. So motor neurons, sensory neurons, each one has a different function, and they connect. In many cases, they connect with each other. Now, how do you connect motor neurons and sensory neurons? You have a connector in the middle. That is the interneuron. From the name, inter means in between. So the interneuron provides the communication between various kinds of neurons, sensory neurons and motor neurons. Now, when we speak about, so here, for example, is a kind of sensory neuron that goes from, it has dendrites that are all embedded within your skin. And as we keep going, it has all these dendrites come into one big cable, and that cable keeps going, and then you have a cell body, and then from the other side you have then another cable or another an axon that is going to travel, and then at the end you're going to have dendrites. They are called telodendrites or synaptic terminals, and I'm going to speak about these later. But this is how the structure is, a sensory neuron. Now look at the motor neuron. The motor neuron has dendrites and cell body, and the dendrites come out of the cell body. And then you have an axon, one single thing that goes down, and at the very end of it there are telodendrites, or you call them synaptic terminals, and they connect onto muscle fiber. So when the signal is processed in the cell body, then it travels through the axon and it activates the muscle fiber to make it contract, to make it shorter. And then on the left you're going to see interneurons, different kinds of interneurons. They are different. When you look at the structure, there are many different kinds, but these are two kinds of them. And they connect one, say, for example, a motor neuron with a sensory neuron, so that what you feel, you can act upon. So now I am going to explain the knee jerk, and I need you to pay close attention, because I'm going to ask you how exactly the knee jerk works, and how the circuit that makes the knee jerk, how it is connected, and how exactly it works, where the signal travels step by step by step. Because this is a process, you really need to pay attention to the sequence. What happens first, what comes next, and so on. So all of us know what the knee jerk, how it works. When we go to our physician and they tap our knee cap here, tendon, and then the leg is going to jerk forward. How does this work? On the tendon that is below your kneecap, there are sensors that are part of many endings of sensory neurons, and they send their axon, the dendrites then make a little cable and they travel, and then they have the axon keep traveling. I'm sorry, one second. It keeps traveling here, that's the blue, and it goes to your spinal cord. So in the spinal cord, they terminate there. That sensory neuron stops right there, connects with motor neurons on the other side. Now, the connection is made by interneurons. So inside of the brains, of the spinal cord, inside, then the dendrites, the telodendrites or the axon terminals of the sensory neuron connect with the dendrites of the interneuron. And on the other side, the axon terminals of the interneuron, these connect with the dendrites of the motor neuron. So this is the motor neuron, and the dendrites here are connecting or synapsing with the synaptic terminals of the interneuron. So this is how this circle is connected. Then the motor neuron on the other side, its telodendrites or synaptic terminals are embedded onto the muscle fibers of the femur muscles and the hamstring, and they make here, they have a neuromuscular junction, and they will release neurotransmitters to activate the muscles of your femur. When they activate, they shorten these muscles. They make them contract. Shortening, we call it contract. All the muscle knows how to do is to contract. So when that muscle contracts or shortens, it is going to pull at the structure that is connected with. And it is connected with the leg, so it is going to jerk your whole leg forward. So I will explain, I explained now how this pathway or how this circuit is connected. So then I'm going to explain the steps of how the signal travels from your knee tendon to the muscles of your femur. So first, the tapping signal activates the dendrites of the sensory neurons that are embedded in the tendon, the knee tendon here, below the kneecap. And the signals then travel along the sensory neuron and keep traveling to go to the spinal cord. In the spinal cord, the dendrites, in the spinal cord, the axon terminals on the other end, the axon terminals of the sensory neuron are connected with the dendrites of the interneuron. And on the other side of the interneuron, you are going to have the axon terminals of the sensory of the interneuron are synapsing with the dendrites of the motor neuron. So I just want to clarify, and I'm going to speak about neurotransmission in more detail. Think about the neuron as a person. The neuron can only take information through the dendrites or the cell body. And then it can also take it through, the synapse, the axon. However, the signal has one way. It only travels in one single way. From the cell body, it travels to the axon terminal on the other end. It doesn't go backwards. It only has one-way transmission from the dendrites and the cell body to the axon terminals, the ends of the axon or the branches. So, and then you can think about your feet as the dendrites. So if you line up 10 people and each one is laying down in front of the other, one person gets a signal, passes it to their feet, and their feet connect with the head of another person. That is the dendria or the synaptic terminals, like the hairs, like the dendrites, and the head is like the body. And then the signal goes to the other person through the body and to the axon to the feet, and then from the feet to the head of someone else. So that is how it goes. So the signal goes to the sensory, to the dendrites of the sensory neuron, and they travel along the sensory neuron, and then to the synaptic terminals of the sensory neuron. Now, the synaptic terminals of the sensory neurons are connecting with the dendrites of the motor neuron in the spinal cord, and the synaptic terminals, or like the feet here in that case, of the interneuron synapse with the dendrites of the motor neuron. You notice that interneurons are found only inside of the spinal cord as a connector to close the circuit. Then, on the other end of the sense of the motor neuron, now we have the dendrites, the synaptic terminals of the motor neuron, red, embedded into the fibers of the muscles of the femur, and they make neuromuscular junctions where they release neurotransmitters. Neurotransmitters bring in the signal that originally came from the sensor neuron. So the signal keeps traveling until it reaches the neuromuscular junction that is made between the axon terminals of the motor neuron and the muscle fibers, and then the neurotransmitter causes the muscle fibers to jerk, to shrink, or to contract. When they contract, because these muscle fibers are connected with the lower knee, then they shorten and they pull the whole leg forward. Because when you shorten, there's, on the other end, the leg is tied to these muscles. They shorten, they pull the leg forward. So that is the sequence exactly of how the knee jerk works. Please make sure to really understand it well, know how it is connected, what the parts that make it up are: sensory neuron, interneuron, motor neuron, how each one is connected with the other, and then how the signal travels from one to the other. In terms of the anatomic structure of neurons, what they look like, how big they are, and so on, there are more than 100 different kinds of neurons based on their anatomy. So we when we explain neurons and show images, we simply show the one that has similarity with most of the others. And that is the simplest form of neurons. That is the motor neuron. So this is a motor neuron. This is a motor neuron. Classically, it has been used because it's a simple form, and it has elements that are similar to other neurons. So we're going to speak about the different parts of the neurons and how they function. So first, a neuron has a cell body called soma, or cell body simply. Soma means body. And inside of it, there is a nucleus, that is, it has the DNA, and outside it has ribosomes, it has other areas where you can make, products like neurotransmission. You can actually make new neurons. Whatever information you have in the nucleus, the DNA, goes outside to be copied, and then that neuron can make other neurons, or it can make neurotransmitters and other chemicals, other elements, like spare parts for the neuron to repair it and so on. On the body, or out of the body, come branches called dendrites, which mean branches. And each dendrite at the very end, it has a little kind of cup-shaped, ending like this, where it can synapse with another neuron to make a synapse like this. So, and remember, one neuron like this can synapse with 10,000, or it can synapse with maybe 50,000 neurons. So it depends on what kind of neuron we're speaking about. But again, we are using, small branches here just to explain the basic idea. So, dendrites, cell body, nucleus inside of the cell body, and there's a little triangle at the end of a cell body. That little triangle is called the axon hillock. That triangle here. That connects the cell body to the axon here. The axon is like a thread, a small thread, but it is actually a tube. And it has fluid, it has a cell membrane, and everything. So the axon hillock, again, it connects the cell body to the axon, And in the axon hillock is the firing station or firing signal. That is where you find the switch that actually fires a signal through the axon. Let's say, for example, that this is the dendrites are part embedded in your skin. And let's say that something pricks you, like a mosquito, biting you. So you get the signal, the pain signal travels along the dendrites to the cell body, gets processed. And then all of these dendrites are going to be sending signals as well, bringing signals from many, many neurons. So in here, in the axon hillock, then it takes these signals and say, oh, should I fire or should I not fire? The axon hillock makes the decision as to if the neuron is going to fire in response or if it is not. So if it fires, the signal then travels along the axon to the branches here that are called telodendria or synaptic terminals or synaptic branches like this. So the signal travels. As it travels, there are axons. They have myelin segments that insulate, like marshmallow pieces, like Schwann cells in this particular neuron. So these segments insulate what's inside the axon from the fluid that is outside the axon. And by doing so, they make the signal go faster, and also the signal doesn't get lost. In between one segment and another, there's a tiny space that is called the node of Ranvier. Node of Ranvier. These little spaces, nodes of Ranvier. So the axon itself is that yellow structure going, and it has myelin segments, and the space between each segment again is called the node of Ranvier. So as the signal goes, it shows up along these nodes of Ranvier, and it shows in one segment, and then as soon as it shows in the other segment, then it goes off here. Like, some Christmas lights, if you have this light goes on, and then as soon as it goes off, this light goes off, and then the next one goes on, and so on. And then the signal goes in one way. It does not travel in two directions. The signal along a neuron goes one way from the body or the axon hillock to the synaptic terminals of the axon. And then from there, they go to the dendrites of another axon and the cell body of another neuron, and the signal travels again the same way like it did with the other one from the axon hillock to the synaptic to the axon to the synaptic terminals, and then so on. So this is how neurons communicate. So again, we explained this, and remember that the axon has insulation. That insulation collectively, all the segments together, sometimes they're called the myelin sheath. The myelin is the substance, like the dough, that the substance itself that you can make different segments of myelin out of. The telodendria or telodendrites or synaptic branches, these are the parts at the end, on the other end of a neuron, and the signals go from here to the dendrites and cell body of another neuron. Like imagine, like I said, 10 people or 100 people laying down, one person here, and the feet, and then after that another person with the head of the second person touching the feet of the first. And then you keep going, and then the feet of this person touch the head of another person, and you keep just going. The neurons will be like this. In the space, so this is a magnified, I'm going to bring you here again. See, if you take one of these dendrites or synaptic terminals, and the end, they end up having a shape like a foot, like this. And if you take the dendrite, back again, the dendrite of, say, one neuron here or here, and at the very end of it, you are going to have a structure that's shaped like a cup. So one, the neuron, the synaptic terminal, is shaped like a foot, and the other one is shaped like a place to hold that foot, like this. Like this. And there's a gap in between them that is called the synaptic gap or the synaptic cleft. So we call this neuron, the neuron that sends in a synaptic terminal to connect with another one. That neuron is called the presynaptic neuron. So the synapse is basically, the area where the two come in contact or communicate with each other. So this is called the presynaptic terminal neuron. And this is called the postsynaptic neuron. It is part of it. So in between the presynaptic terminal here and the postsynaptic dendrite here, in between we have that gap called the synaptic cleft, and the neuron, the presynaptic neuron, is the one that brings in a signal from its axons, brings in a signal, and then the one that is the postsynaptic neuron is the one that is receiving the signal. How do they communicate? They have in between them, they have that little gap. The presynaptic neuron synaptic terminal comes in, it has little bubbles, we call them vesicles, that have neurotransmitters. So these neurotransmitters, bubbles or vesicles, come down, and when the signal is coming in, these vesicles fuse with the wall of the synaptic terminal, with the walls. As they fuse, they dump their ingredients, the neurotransmitters, in the space, in the synaptic cleft, without opening the gates. They only allow the synaptic vesicles to fuse and then to dump their ingredients without opening the membrane of the presynaptic neuron. So then the little vesicle walls fuse to be part of the wall of the presynaptic neuron, and then the neurotransmitters float in the space between in the synaptic cleft. So watch, for example, for these brown neurotransmitters here, whatever, that's like calcium. And then they go on the other side of the dendrite. There are channels. These are channels, and each channel has like many places on its its surface, and each neurotransmitter is shaped like a certain shape that can only fit on a particular site on that channel. It's like lock and key. When it lands in there, it docks on it, binds, then the gate either opens or closes. So that's what a neurotransmitter does. When that happens, then that opens the gate, for example, to bring in the signal, and then the other cell is going to go bring it to the cell body eventually, and then to the brings it to the axon hillock, and then to the axon, and so on. So the neurotransmitter opens gates to send in the signal, or it might cause the gates to close if the signal is to say to the neuron, do not fire. It's an inhibitory. It means stop. So depending on the activity, the neurotransmitter can open channels, receptor channels, or can close them, depending if you want the signal to go into the next neuron and to be fired and so on, or to be just terminated So I'd like you to know what the synapse is. A synapse is a point of contact between one neuron and the other. It is made by a by a synaptic terminal that comes from a presynaptic neuron. So presynaptic means the one that brings in a signal. And between the dendrite or part of a cell body of a neuron that is receiving the signal. And then there's a gap in between that is called the synaptic cleft. And the presynaptic neuron has vesicles that hold neurotransmitters, and these neurotransmitters are dumped into the synaptic gap, and then they bind to special receptor channels that are embedded on the membrane of the postsynaptic neuron. And then the signal, then if it is a signal to activate, then it will be processed. It will travel to the body. It will be processed there, and then goes to the synaptic, to the axon hillock, and from there to the axon, and from the axon to the axon terminals or synaptic terminals. So the process here is explained again in detail as I explained it. And if you have any questions, please write them down and ask me. I'll be happy to explain this again. So here again, as I explained, let's say let's say a signal is coming in. I'm gonna use this time the mosquito analogy again. So you have in your skin, you have all these dendrites embedded in the skin. And then, say, a mosquito bites you. The signal then travels along the sensory neuron in one direction. It goes from the dendrites to the bodies of the sensory neurons. And then as it goes from the sensory neuron, it then comes to the synaptic terminals. Hold on one second. It goes from the dendrites to the cell body to the axon hillock, and the axon hillock might go say, that is a significant kind of signal. I am gonna do something about it. So it is gonna fire a signal, and the signal travels along the axon. The way the signal travels along the axon, it looks like as if it is jumping. We call that saltatory conduction. So because it goes from one node of Ranvier to the next quickly, and that is called saltatory conduction. So then it goes to the synaptic terminals of the sensory neuron, and when that signal then travels from the synaptic terminals to the dendrites of another neuron, say the interneuron, for example, in that case, because you want to move your hand. And then that interneuron is gonna process the signal in the same way like the sensory neuron did, from the dendrites to the body, to the axon hillock, through the axon, and then it refires the signal, sends the new signal again through the dendrites. And then on the other side of the interneuron, you have a motor neuron that is connecting onto your arm, and then you just go and withdraw your hand and take your hand away. When I speak about a sensory neuron, I'm not speaking about a single one. I'm speaking about millions and millions and millions. I'm just saying just one as an example. There isn't just a sensory neuron. There are millions of sensory neurons embedded on your skin that enable you to function. So again, that is so be able to answer a question like, say, a mosquito bite. How does the signal travel from there and to the end when to cause you to take your arm away, your hand away? What goes in between? How does the signal travel from a dendrite of a neuron to the axon terminals, for example? So signals that excite another neuron, that activate channels and activate receptor channels of another neuron are called excitatory. They cause firing, and these signals, as they travel, they cause the neuron to refire the signal. If the signal is to prevent firing, we call this inhibitory. So inhibitory, again, neurotransmitters cause the next neuron not to fire. The firing, when the little electrical spark is fired as the signal travels from one side of the neuron to the next neuron, we call this the action potential. So the action potential is the firing that the neuron performs in response to some kind of a stimulus or a signal coming from another neuron or from outside of the body, for example, or inside. So the action potential, it either happens, the firing either happens or it doesn't happen. So it reaches a certain point. Say, for example, there's a certain point. If the stimulus or, the signal that is coming in is significant enough, it is gonna just cause that neuron to fire. It is like the trigger of a gun. Say, for example, when you pull, you don't just pull and then the thing fires. You just keep pulling, and when it reaches to a certain point, there is no way to go back. It just fires. So that's called the threshold. So this is why I say an action potential is an all or none event. That means it either happens or it doesn't happen. But when it happens, it comes in one size. That's it. It doesn't come like a big action potential and a small action potential. It is just one size, one firing. So as we approach wrapping this up, we speak about some concepts, and we speak about just highlights of development of the nervous system. The structure of the nervous system, it begins actually seriously starting to form is in the third week of gestation, of pregnancy, third week. And at that time, the ovum, the fertilized egg, it has 3 layers. So if you look here, there's one layer on top, and there's another layer and another layer in here. So you could see here, 1 layer, 2, 3 layers. And on top of the ovum or the fertilized egg now, you start to have like a furrow, an indentation, like on the very top of it, of the cell, like this, in the back of it. That furrow then becomes deeper and deeper and deeper, and it goes down, and eventually, as it goes down, then the 2 sides on top, on either edge, this edge and this edge, they come close and they close, and they form a tube underneath them. That tube is called the neural tube, or the neural tube. The top of it here is called the neural crest, but the tube is here. Now, this tube now just goes from all along, from the beginning of the cell, from the back of it, the beginning to the end. And basically, that's what makes the spinal cord. Think about the spinal cord. And then the brain connected to the spinal cord. So, in there, the neural tube is where all neurons are born. And glial cells and everything, these are born there. And then they now have to travel to their destination. So the birth of neurons at a fast rate and the spreading of neurons and brain cells is called proliferation. The stage where neurons are born at a very, very rapid rate and they spread around. And they are born at a rate of 250,000 neurons per minute in one single minute. Because now that by age 6 months, during 6 months of birth, that brain has to have 200 billion neurons. And you need to build this in 6 months. So the rate, you can imagine, 250,000 every single minute. So now all of these are born here, and they have to travel now to their destination. What is the name of the glial cell that guides newly born neurons to their destination? So you should know that. So then the migration process, it enables, through this process, enables the cells to travel from the neural crest to their destination, whether they are part of the auditory system, the motor system, whatever you call it, they will travel there. And they use neuroglial cells so that they can be guided and they receive nutrition. By the time they reach their destination, they are fully grown and they are ready to function. If there's anything that disrupts the migration process, that is going to lead to either death of the fetus before the fetus is born, or it might lead to significant brain disorders. Now we're going to speak about basic concepts that have to do with this part. So when we speak about, say, a nerve, what is a nerve? The nerve is basically a bundle of nerve fibers, of axons, that come together, and they are like little threads, are bundled together and into one bundle, and they have a wrapping around them, some kind of connective tissue that wraps them into a hose, like kind of that. And inside you have all these axons traveling from one side to the other, and you have also blood vessels that travel with them, and you have different, pathways to nourish the axons and to enable them to perform their functions. So that's what a nerve is. Gray matter is, let me go here. Gray matter is the area where the bodies of cells reside. The cell body of a neuron is filled with oxygen, with glucose, with nutrients, and it is pearl gray in color. And these usually reside, the cell bodies, 80% of them reside in the cerebral cortex, the very, very top layer of the brain that is about 4 millimeters thick, and it surrounds the entire brain. So that is called the cerebral cortex. You can see this line here, and that little dark brown, that is the cerebral cortex, and this is another name for it, is the gray matter. So gray matter also exists in the spinal cord as well, that outside layer like that. So the cell bodies, let's say here, they reside in the, mostly inside of the gray matter. They send down their axons. The axons are like little threads. What are these axons? What are the elements that wrap around them? We call that myelin, that white substance. It is fat, a kind of fat. And Schwann cells and oligodendrocytes are the ones that provide myelin. So myelin is white. It is a kind of fat, and this is why all these hundreds and thousands of these axons travel down below the gray matter. We call that the white matter. All of this is white matter. All of this is white matter. So all like the white matter from one hemisphere of the brain comes together and gets bundled together into a gigantic cable that you could see here. And that gigantic cable or pathway, highway that connects right hemisphere with left hemisphere is called the corpus callosum. That corpus callosum, you see part of it here, but if you see it from above, it will be like this. Like this. And all the axons from one hemisphere to, they basically go and connect with the axons of the other hemisphere, and we have now one whole brain. So remember again, the corpus callosum is the biggest commissure or the biggest highway that connects the right hemisphere with the left hemisphere. So now you know what the gray matter is, what the white matter is, what a nerve is. A fasciculus, another name for it is a pathway or tract or track. So all of these are different names for a pathway in the brain. And these are basically bundles of nerve fibers that specialize in one particular function, and they transmit information from one side of the brain to another. The biggest pathway in the brain, or commissure, also as another name, commissure, is the corpus callosum, and it connects right hemisphere with left hemisphere. Then the neuromuscular junction. The neuromuscular junction is the site where muscle fibers here connect with neuron axon terminals. If you look inside, axon terminals have many channels, and they kind of line up with muscle fibers. Muscle fibers have channels, and inside the neurons bring in neurotransmitters, and then they activate the receptors channels along the muscle fiber, and then that is going to cause the muscle fibers to contract, to shorten, and that will move whatever structure that is tied to the muscle is going to be moved. So now we have concluded the discussion of the cellular components of the nervous system, and in the next few minutes I'm going to tell you the questions that you will expect and that you should be able to answer if you have a problem. Somehow you see that after you took your notes and after you listened and viewed this lecture that you still cannot answer these questions. I would recommend that you go back and listen to the segment where that information is discussed. If you still find it difficult, then please bring the question to the class. So here are the questions that that could serve as the guide for this lecture, and any question should be within what I'm speaking about here, within one of these questions. I mean, any question that comes up from about this part of the lecture in the exam is going to be one of the questions I'm going to tell you. So, what are the general functions of glial cells? List at least 7 general functions of glial cells and explain. Like, you don't say insulate, you just say insulate the axons of neurons so that the signals travel effectively along the axons. Don't just list things that I'm mentioning in the slides. The slides are just, they have information to help us be organized. But for purposes of exams, you need to explain. You don't just memorize things on the slides and spit them out. That's not the idea. What are the major differences between neurons and glial cells? Explain how astrocytes support the structure of neurons. Explain how the blood-brain barrier is formed, what kind of cell forms it, and explain how the blood-brain barrier works. List the 4 main functions of astrocytes. What are Schwann cells? What do they look like? What is their function? What is their function? And what part of the nervous system do they exist in? What do they look? I mean, how many Schwann cells are needed to insulate one neuron? For example, do you need multiple Schwann cells to insulate a single axon of a single neuron? You do the same for the oligodendrocytes. Where do they exist? And how exactly they are structured? What kind of neurons do they make the myelin for? Microglial cells, what are their functions? Radial cells, what do they look like and what are their functions? Speaking about the anatomic structure, how many types of neurons are there? Now, speaking about the physiology of neurons. Based on physiology, how many kinds of neurons are there? I hope you say 3, because there are only 3. What is another name for sensory neuron, for a sensory neuron? What is another name for a motor neuron? What are the functions of the motor neurons and what are the functions of sensory neurons? What are the functions of interneurons? Explain exactly and in sequential steps the knee jerk and how it works, step by step by step. Say how the circuit is connected from sensory neuron and connecting with interneuron in the spinal cord, and then in the spinal cord, the interneuron connecting with the motor neuron, and on the other end, the motor neuron connecting with the muscle fibers. And then you explain how the signal goes from the anvil as it hits your tendon back to the muscle that makes your leg jerk. What are the parts of the neuron? So know every specific part and know how the functions, the particular functions of that particular part. Be able to identify different kinds of cells on images. Be able to identify different parts of neurons and structures on images, so that you can identify them and know how to answer questions. What is a synapse? What are the parts that make up a synapse? And how does a signal travel across the synapse? So in that case, the signal goes from the synaptic, from the pre-synaptic axon terminal to the membrane of the postsynaptic neuron. So you speak about how the synaptic vesicles, the neurotransmitter vesicles come from the presynaptic neuron, how they fuse along the walls and dump their ingredients in the synaptic cleft, and how these neurotransmitters then bind onto the receptor channels of the postsynaptic neuron. So you need to study this several times so that it can make sense and that you remember the information. Explain how, what is a presynaptic neuron? What is a postsynaptic neuron? Explain how a signal is transmitted along a single neuron. So, for example, using the mosquito analogy, the mosquito bites you. What happens from that point on? How does the signal go from the dendrites of the sensory neuron to the body, to the axon hillock, to the axon, to the synaptic terminal? So these are the steps when the signal travels within a single neuron. When it goes to the synaptic terminal, all you say is it just goes out then to other neurons. That's it, within one single neuron. If I ask you to explain a circuit, for example, what happens from the time that the mosquito bites your hand, your skin, to the time that you move your arm away, that is exactly the same as explaining the knee jerk. You explain sensory neurons do this, and then interneurons do that, and then motor neurons do this, and then your hand is pulled away from the source of pain. What is the neural tube? What is cell proliferation? what explain the process of cell migration, what kind of microglial cells, what kind of glial cells aid or help in the cell migration process. How do they do that? You need to know what a nerve is, what gray matter is, what white matter is, what is a fasciculus or pathway or commissure, what is the biggest commissure in the brain, and how is it made. That is the corpus callosum I'm speaking about. And explain also what the neuromuscular junction is. So that brings to conclusion all the questions that you need to answer from the lecture that I have just recorded. All these questions are the questions that will serve as material for the exam on this particular lecture. So again, if you can't answer any of them, or your answer is not absolutely 100% perfect, then you need to go back and review. How do you know it is perfect? So you take these questions, answer them out of your own memory, and write them down. If you can't, or if you feel there's any problem with the question, parts are missing or the sequences are not correct, go back again to the segment of the class, of the lecture, where I discuss it, and then listen again, and then rewrite your answer again until you are able to write it independently out of your own memory. And then when you do that, then the answer, you own it, it becomes part of your knowledge, and you will remember it for a long, long time. That's the right way to study. So thank you, and please bring any questions that you might have to the class.